Our next session is our bookend, our statistics bookend, where we will get down and dirty with the statistics questions. James, it would be helpful if you could kick us off here.
Wonderful. Thank you, Sarah. So it's my pleasure and privilege to be presenting on behalf of the excellent statistical working group, the members of which have provided quite a bit of thoughtful input and feedback on this presentation from their various perspectives. We're going to be talking about multi-organ system composite endpoints, which we of course could not do without input from all of the different organ-specific working groups. So I'm also quite grateful to all of the working groups for the output they've been providing, and to AHRQ and FDA for creating the structure that can make this kind of work possible. So what I'm going to walk through today is our approach to creating and validating a multi-domain endpoint. We're going to talk very briefly about the process, and then get to some straw man endpoints that we're going to seek feedback on in the Q&A session. So whenever we create an endpoint, we have to be thoughtful about the context of use. And that's even more true for a multi-domain endpoint. There's going to be hundreds of decisions we're going to have to make in the construction and validation of this endpoint, and those all need to be informed by a particular view of for whom this endpoint is going to be used and what value we hope it to add. So for this exercise, drawing on the work of the working groups, we're going to focus on really plasma cell directed therapies for a broad population, hopefully where we don't have to exclude patients often based on their level of organ involvement. And we'll also be thinking about endpoints that would be suitable for full approval, since that seemed to be the area with the kind of the most potential added value. And so in this context of use, the value of developing a composite is really by tying together all of these outcomes across organ systems. If we really have a therapy that is getting at the root cause of the disease, then we should be seeing these multi-domain impacts. And the hope is that by pulling those together into a composite, we can rigorously measure the effect of treatment, but do that sooner than if we had to wait for major organ damage to occur or mortality to occur during the trial. So as we think about composites and how to pull together the information, it's really this context of use and this ultimate goal that we have in mind. And so the overall process is, we're sort of sitting at this yellow triangle here where the working groups have identified organ-specific endpoints and measures. Still some work to do there and things to validate, but a lot to work with already. And where we're sitting now, we need to pull all this together into some candidate multi-domain endpoints and then go and validate those in data. So as we start this process of developing candidate composites, it's helpful just to touch on some of the considerations to have in mind when developing a composite. So number one is there's important pitfalls to avoid. There's all these good things that can come out of a composite, but if we're not careful, we can fall into some traps. Lack of interpretability is a big one, especially when we're combining across multi-organ systems. We wouldn't want to get to an endpoint that in the end, no one's going to know what it means or how to interpret effects on that endpoint. The other thing to keep in mind is the sensitivity to drug effects. It can sometimes feel like the more we add to an endpoint, the more chance we have of detecting a benefit, but we also have to keep in mind that as we add up different domains, we're also adding up the noise across those domains. And so keeping an eye on the kind of balance between hypothesized signal and noise is going to be important. Another thing to keep in mind that can really help frame some of the discussions around composites is they're not the end all be all. We're not trying to come up with one endpoint that will answer all the questions. We're always going to be looking at the totality of the evidence. And so the the real question that we have when thinking about what to put into a composite is really, do we create value by combining these things into a single measure, right? Because we're always going to have the value of each one individually. And so it's often the case that there might be a measure that's super valuable on its own, but there's a reason not to include it in a composite because it's not going to create value by combining it. It could create confusion. So with those, we're going to walk through some of the decisions that we came to that are kind of foundational to putting together a composite and really allowing us to focus on something that's going to be achievable and we think valuable. And so one of the things we have in mind as we start down this path is just an understanding of how the different domains fit together. This figure on the screen is obviously oversimplified, but key points are that hematology is really underlying everything here. And then each of these organ systems are having impacts on overall quality of life. So with that, here are the kind of foundational enabling decisions. And I'll walk through these, and I would invite everyone, if you have questions or comments about these decisions, to please submit them as soon as you have them, rather than waiting until the end, and that will help us kind of calibrate when to go to the open up the Q&A session. So the first decision we made is to focus first on end organ impacts. These have the strong rationale for a composite. If a therapy is effective, we'd hope them all to move in the same direction, improving eventually at least. And we know it's a progressive disease without treatment. Hematology is, of course, incredibly important. And ultimately, we might combine an organ composite with a hematological endpoint. And I think Dr. Dispenzieri gave some excellent examples and thinking around that in the first presentation. But our thought is, let's focus first on pulling together the end organ information and as a first objective. The other thing that we want to do is focus on measuring clinically meaningful outcomes sooner rather than later. So for example, if we can measure a clinically meaningful impact on kidney function through eGFR or proteinuria, let's try to do that rather than taking the longer term milestones such as end stage renal disease. We also thought it would be important to ensure that each component in our composite is itself something that could serve as an approvable endpoint. There might be reason and arguments that one could make to relax that, but we feel like as a starting position, this is an important place to start. Let's see how far we can get following this principle, because it helps ensure that any effect on the composite is then going to be itself clinically meaningful. If we start including biomarkers that are not yet accepted as surrogates into the composite, then we can get into situations where the interpretability of the composite itself is not robust. Another decision we came to was to reserve the general quality of life measures really for validation purposes, rather than trying to include them in the composite. And this really comes from that overlap. We'll talk later about kind of domain-specific quality of life, but for overall general measures of quality of life, if those are validated by and approved by the HRQL working group, those would serve best as kind of anchors or reference points against which we could compare a candidate composite outcome. Whereas if we included them, that overlap with all the domains could make things difficult to interpret. Okay, and then three other decisions, and these I think grow out of some of the presentations and discussions that you heard today. First, we felt that GI outcomes weren't yet ready to be included in a composite. There weren't measures that were available for AL, and also with the kind of responsiveness to certain background therapies, it wasn't immediately clear that that GI outcomes would add more signal than noise to a composite. We're also not able to include liver outcomes at present, at least not without more evidence. And then finally, domain-specific quality of life. We suspect there would be quite valuable outcomes to include here that will probably come out of the work that Vaisali just outlined. Ideally, these would represent things like liver, pain, fatigue, mental health, domains that are not already well represented in the other endpoints that we're considering. So with those kind of enabling decisions and focus areas, we can now start creating some candidate composite outcomes for AL. And as I mentioned at the beginning of the workshop, there's different kinds of composite outcomes, and they all have different pluses and minuses. And so we're actually trying to create versions of each of these types of composites for AL, drawing on the different measures and domains that the working groups have identified. So they each have their pluses and minuses, and we're going to talk through those in detail as we go through each of the composites. So the first step is to identify the different domains and measures that we want to include. So even before we say, okay, decide whether we're going to have a response outcome or a progression-free survival or a multi-domain responder, we want to narrow down to what are the specific measures and thresholds for clinically meaningful worsening and clinically meaningful improvement that we're going to consider. So based on the output from the working groups, we've started to kind of frame up what the components of a composite might look like. This is very much under construction, and there's decisions here that need to be tested against evidence. But these are the ones that kind of seemed most ready to help us start thinking about potential composites. So we have, for cardiac, we selected the 6-minute walk distance with a 33-meter threshold. We could revisit that and say, should it be KCCQ? We might get some input on that from the quality of life working group. For neuropathy, kind of consistent with trials in TTR amyloidosis, we've thought about an MNIS plus the Norfolk, and there are thresholds available for those. They haven't been validated yet in AL, but it seems clear that we'd want a combination like this to represent the neuropathy domain. Similarly, COMPASS, as was discussed, stands out as a very attractive endpoint, but still needs some validation in AL. And then in renal, there was some discussion of combining EGFR with proteinuria to come up with a threshold of worsening and then a threshold for improvement from baseline. So I think all of the thresholds and decisions here need to be worked out, and that'll be a continued discussion and informed by evidence. But we wanted to share this to give a flavor of what a composite outcome might look like in AL. So now I'd like to put forward some straw men for the different types of composites. And I'll go through these briefly because I think a lot of the key issues will probably come up in the discussion, and I want to leave room for that. So one would be a measure of organ progression-free survival. This is very much in the spirit of the MOD PFS that was used in Andromeda, but seeing, can we take some of those organ deterioration measures and make them things that are going to occur earlier in the disease course rather than later. So this is attractive given the precedent in Andromeda and in hematology in general for using PFS type endpoints. The main drawback is that this outcome by itself would not be directly sensitive to improvements from baseline. Obviously, if the improvement serves to delay progression, it would be sensitive to that, but not directly sensitive for improvement. So another straw man we considered is for organ response. So similarly, we would combine these domains and have some logic for what constitutes an organ response. For example, improvement in any of them without worsening in any. But again, that logic would have to be worked out and ultimately tested against data. The advantages of a response outcome is it would be sensitive to improvements, especially in the near term. But of course, it's not looking at delays and worsening. We'd probably still want to look at things like duration of response, but it wouldn't look at avoidance of progression directly. The other thing to consider about response and progression outcomes is they could also impact the inclusion criteria that would be appropriate. In particular, if we're looking at response, we'll only be able to see that in a domain if the patient already has some level of impairment in that domain at baseline. And so we'd have to think carefully, and also data can help guide us in this, of how heterogeneous a population would be suitable for looking at response and for looking at progression. We also considered a multi-domain responder index. The advantage of MDRI is, of course, that we can now be sensitive to both improvements in domains and worsening in domains. So if we're in a situation where in natural history, all these domains are worsening, but with effective treatment targeting the root cause, they're all improving, then using a multi-domain responder should maximize our sensitivity to see that type of effect as early as possible, compared to looking at just improving or just worsening. But of course, the challenge of a multi-domain responder, and this is a slide from an FDA presentation, but I think it summarizes it quite nicely, is we have to be really thoughtful about the measures that go into it, and especially the symmetry of meaningfulness between worsening and improvement, not only within domains, but also across domains, right? So you get into a situation where, if an individual patient's 6-minute walk is getting worse, but their EGFR is getting better, are we sure that we're weighing those appropriately? But again, if everything tends to move in the same direction within patients, then these can work quite well and maintain interpretability. We also thought about the Finkelstein-Schoenfeld approach, which was, of course, successful in the TRACK trial for cardiomyopathy. And this one, we thought about it kind of requires a hierarchy on the endpoints that are included. And what we thought about is using survival at the top of the hierarchy. And then if patients are not differentiated on survival, using something like a multi-domain responder to further differentiate that. And the reason for that is it wasn't clear how to put the organ systems into the type of hierarchy that we thought was needed for Finkelstein-Schoenfeld. I think it's possible to say, which organ systems are less involved or more less important. But in the Finkelstein-Schoenfeld approach, the hierarchy is quite strict in that if we see an impact on things at the top end of the hierarchy, then we don't look at anything below at all. And so that seemed difficult to get to with the different organ systems. And so we propose using a multi-domain responder to resolve that. So before we go into questions, I just want to make clear that this is very much a process. The step we're at now is getting feedback from this workshop and from the follow-up conversations to really finalize some candidate composite endpoints that we think are worthy of testing out in data. And then we're going to develop a plan for doing those data-driven validations. That plan is going to draw quite a bit from guidance that's been issued by FDA, EMA, ESPOR, and others on how to evaluate endpoints in general. And then we're going to go off and look at that data and see which of the candidate endpoints perform well. So thank you. I'll close there and open it up for questions.
We have one from Brendan Weiss. perhaps he could start us off on this.
Sure. My question was, why was NT-proBNP not included in the cardiac domain? And I have a question I was just about to type in related to that is, how do these methods handle the fact that not every patient is valuable for that domain? So, you know, if 70% of cardiac and renal, but only 10 to 20% have neuro and 10% have liver, you know, how do the statistical approaches account for that?
Yep, great questions. So first I'll speak to NT-proBNP. I think two considerations there. That might be great to include. I think the reason we haven't included it yet is because it's not yet accepted as a surrogate outcome. But I'd also say if it was accepted as a surrogate outcome, I think there's a question of maybe we just need NT-proBNP, right? I think others have spoken to how it connects to certainly to cardiac and renal. And so I think there'd be a legitimate question there, which could be answered with data of whether NT-proBNP by itself is sufficient or whether a further composite would add value. I don't know if others from the cardiac working group want to say any more about NT-proBNP.
I agree with what you said. I think I could see how your group would come to the conclusion at this point that regulators would accept a 6-minute hall walk. I think this feedback from the working group would be that there's only one series so far published by the National Amyloid Center, and data suggests that it doesn't have an early effect. So I don't know how much we're going to accelerate development by looking at an NT-proBNP, because we're going to have to wait. Clearly, it looks like from the emerging data to at least a year's time. So I think there's, to be honest, more robust data for NT-proBNP, to Brandon's point, than there is at this point for 6-minute hall walk in AL amyloid.
I guess I would throw back a question. So what's it going to take for it to seem validated? I mean, you know, because there's a lot of retrospective data, there's emerging prospective data from trials, and it just feels like there's like no right answer, you know, there's just no way it'll be accepted, or, you know, I just still don't feel like we have that goal, you know, like where do we go to with the NT-proBNP to satisfy things?
Yeah, maybe Preston, would you be able to comment on that?
Sure. Excuse me. I think that people have to keep in mind, from our perspective, what we see are the large heart failure clinical trials coming in, understanding that these are cardiomyopathies and coronary disease for, you know, and the cardiomyopathies can span a lot of different causes. But still, when you look at least trials of HFrEF, the BNP result from those trials has not been consistent. And I point out that in our mistake with flusequinone years ago for heart failure, a drug we pulled off the market, the NT-proBNP went down as mortality in the long term went up. And so that's what's given us difficulty with that outcome in heart failure in general. I do think this is a unique case for AL amyloidosis, though. Particularly, a strong one could be made for specifically the group of patients that are receiving an experimental anti-plasma cell therapy. Because from Dr. Mauer's earlier presentation, you know, one of the things we have to be convinced of is the biomarker you're measuring is on the pathway in the pathophysiologic pathway. And I think there's a reasonable argument to be made that when you're treating toxic light chains and then getting rid of toxic light chains, that argument could be made fairly reasonably. And last time we talked about this, the FDA and others, we weren't really thinking and separating out amyloid therapies from anti-plasma cell therapies. They were all kind of getting mixed up together in the conversation. That made that difficult for us to get from here to there. But I do think that it's a conversation worth revisiting, specifically targeting that topic of is that a worthwhile thing as a surrogate when you're looking at anti-plasma cell therapies. Now, the next question obviously is going to get to be what makes it better than the hematologic components of an outcome showing a complete hematologic response. I mean, if you measure that your NT-proBNP goes down, who cares if you've had this incredible hematologic response with all the markers that you heard about that Dr. Dispenzieri was talking about. So I'd be interested in her thoughts on this, because in the anti-plasma cell world, I can see how we could have that conversation. But then, we would be asking, what does it add to the hematologic outcomes in anti-plasma cell therapy?
So I want Gianpaolo, please, Gianpaolo.
I think that it adds a lot, actually, because what really counts is the cardiac response. And reducing the light chain does not necessarily translate into improvement in NT-proBNP if your cardiac damage is too advanced. So as Angela has shown recently, having NT-proBNP as a single marker above 8,500 is enough to identify you as a poor prognostic, and is the only criteria we use to stratify. And in my view, NT-proBNP response is enough. We don't need this multi-organ, multi-criteria. I mean, if you follow NT-proBNP, that is enough to provide you significant survival benefit. And I would like also to add that NT-proBNP is the only biomarker extensively validated. We don't know anything about the walk time. I mean, the 3-minute walk or whatsoever. There is one single study showing that, but we don't know exactly how it is sensitive to the response of the patient. Why we know a lot about NT-proBNP. So we have an amount of data that no other biomarker so far has. So the evidence is there. And Preston, I think that I understand that in other cardiological settings, NT-proBNP could have not performed well. But if we think about the amyloidosis and the mechanism, molecular mechanism of this disease, NT-proBNP is certainly suited to be the surrogate biomarker.
Gianpaolo, one question I have specifically then is, what do you think about a trial result, a hypothetical trial result, where you got a complete hematologic response with current criteria that you and Dr. Dispenzieri have talked about, but you didn't really get an impressive NT-proBNP response, versus a trial where you get a great NT-proBNP reduction with no hematologic response? I mean, do you see that span of outcomes?
No, Preston, that cannot happen. We have never seen a really very good cardiac response if you don't have free light chain response. That is impossible.
Okay, so basically then that gets to my point. What is the incremental information? Because if we can make the argument to the regulatory agencies that this is ready for prime time for the anti-plasma cell therapies, which I would start there first, because I think the argument on the anti drugs is going to be tougher. But say we start there, what would be your answer to the, and you and Dr. Dispenzieri, I'd be interested in both your comments about what incrementally does it add, since what you're telling me is you never get an NT-proBNP response without a hematologic response.
Yeah, of course, hematological response is the prerequisite. But if you would like to see how the hematological response translates into patient benefit, then you need NT-proBNP.
Okay, so what you're proposing then is potentially a secondary outcome that the company could base a claim on for cardiac improvement.
Yes, right.
Yeah, which would, we believe, robustly translate into a survival benefit.
Because we know, we can show you data, retrospective or part of trial. If you have that patient, two patients who get an equal hematologic response, but one's NT-proBNP goes down and the other doesn't, you're really worried about that other patient. Now, maybe they need a heart transplant, maybe, whatever. It really, it tells you a lot. You just know what's going on, that something's not good here. And so it has a lot of meaning.
And that's important for us to hear that. And Matt, from a cardiology point of view, I'd love to hear your thoughts here. But I think that the regulators, at least at FDA, would be open to hearing this and the importance, Angela, of what you just brought out, and Dr. Merlini, what you just pointed out. Matt, would the community be supportive of exploring this in cardiology?
Yeah, I think to your point, an excellent one. I live in both worlds, heart failure and amyloid. I would not walk into the FDA and endorse the idea that we should be using NT-proBNP for any disease in cardiology other than AL amyloid at this particular point in time. But the evidence is in contradistinction to what we all know in heart failure. BNP can go in any different direction. We can have a billion different outcomes. It is very robust and highly predictive of what happens to patients in the long term. So I don't think
Giovanni Palladini's first paper did look at that, that cross between hematologic response and NT-proBNP. I'm trying to think if there are other papers that have done that exact same analysis, but it certainly can be done to show that differential benefit, that there is an interaction. Every multivariate that's done that shows, independent is essentially doing that. It's not practical, but it's done all the time.
There are people showing that adding NT-proBNP benefit clearly adds an important, as clinicians, we know that if you do not reduce NT-proBNP, you have to look about something else because the patient is not going right. So it's really an instrument that everybody working in amyloidosis is using daily, and it provides certainly additional information. Renal response does not correlate with survival. So that is about these multiple domain criteria. Renal response does not add to survival. The only one that adds to survival is heart. And one anti-amyloid therapy clearly was stopped. And I was sorry about that because it was not able to show heart benefit. And if you don't show heart benefit, we consider one drug ineffective in amyloidosis.
I think we also have a comment from Melanie Blank. Would you like to add on here?
Yeah, so I was just thinking about the can you hear me?
Yes.
Yeah, the usual way that we evaluate an endpoint for accelerated approval, which would be a typical thing for a biomarker, NT-proBNP in this case. And so what we like to see is that it's reasonably likely to predict the clinical outcome of interest. Here would be some cardiac outcome.
Yeah.
It sounds to me like you may
honestly
support that.
That's a pretty good outcome.
Okay, so let me just finish what I'm saying, okay? So I think it sounds, that you may have the data to gather a package to support that. And then, I don't know how that would go. Maybe James can address that, how that would get incorporated if it's just sort of an accelerated approval. you know, if you went on that endpoint more than some of the others, that gets a little complicated. But I think accelerated approval would be you know, it's basically an approval. So it might be worth considering going down that pathway. But again, you need to have the data to support that it's reasonably likely to predict a cardiac outcome.
Yes.
I just wanted to follow up on both Preston and Melanie's point, which is I think we would benefit. We believe we have the data, but it may be that we have not organized it, presented it in such a way that appropriate regulators would look at it and like we do very favorably. Someone asked, I think in one of the questions, which we didn't touch upon, how good is this as a biomarker to predict what's going to happen, for example, to an individual patient? You know, what is the sensitivity and specificity of the changes that we've utilized, which we can show markedly different survival curves, but how good is it? I believe the data is robust, but it would be helpful to garner expertise from FDA, EMA, and others in trying to evaluate this data more carefully, I think, because we believe it's the important pathway forward at this point in this particular condition, I would say, as a cardiologist.
That brings us to Joel's questions about, you know, patients who aren't getting plasma cell directed therapy. So Joel, I think you had a couple of questions.
No, I think a couple of things. I assume that any composite score or standard would be weighted relative to those organ involvements that have already been shown to have a critical impact on survival. So I assume that's built into the mix. But the question that you referred to is really this notion of we now, you know, have the precedent in TTR of being able to stabilize a protein and get a good clinical response. So the question is, in those patients who have hematologic responses and very good ones that don't get good organ responses, and one decides to go the protein-directed therapy, how do you measure those? And do you use the same sort of composite scale that you use for the cytotoxic therapy? So I think that that's a consideration that's now sort of coming to the fore as a number of laboratories and companies are developing these sorts of agents that are really not plasma cell directed. I think that that's something that has to be thought about now. You know, I just think that the people die because they have organ failure for the most part. And the distinction between complete hematologic response and organ failure, that's a major therapeutic challenge. And for the moment, that doesn't appear that it's going to be, I think it will be improved, but not eliminated by the current agents that are being introduced. So I was curious to hear how, as most people know me as a TTR guy, but I have a long interest in AL, and we were very interested in this problem at the time and interested in the context of cytotoxic therapy. But I think that it's now clear that that's not the answer, or not the whole answer, and one has to be able to address it both in the context of being able to predict it and then address it.
Yeah, so your point is that in this way you stabilize the protein and you reduce the protein toxicity, right?
Right.
So your endpoint is organ improvement, right?
Right.
And then NT-proBNP is the most effective biomarker to measure organ improvement we have. So if your compound improves NT-proBNP, is working. If it does not, it's unlikely that it will work.
I have no doubt that any study should include proBNP. I mean, I could go back to I think that the issue that was alluded to a little bit before by Preston is that I don't think any of us who worked with amyloid heart disease of either type were actually convinced that the usual criteria for congestive heart failure efficacy were going to be valid in the amyloid population. Just because amyloid is a specific disease with an etiology, and heart failure trials generally encompass a large number of etiologies, and you're really looking at the end stage when you had no chance to look at the etiology.
Yeah.
So,
Thank you very much, James.
So just to wrap up, maybe I'll wrap up and answer one of the outstanding questions that I think was posed first, which is, patients are heterogeneous. They're going to be in different places at baseline. Some may be able to respond in 2 domains or 3 domains or none. And how do we handle that? And I think that question kind of gets to the heart of why a multi-domain composite would be valuable in what is truly a multi-domain disease. I think we're very used to focusing on one domain at a time, and a lot of the kind of precedent is geared towards that. But in a multi-domain disease, that means we might have to limit enrollment or measure things that are not really getting at the full value for the patient. So there's, I think we need to develop the candidate endpoints and put them to the test by using the evidence, looking at data, and see if they are interpretable and if they would increase power. But it is absolutely the case that patients are going to be heterogeneous in the degree to which they respond or progress and the number of domains that are affected. But that's kind of the point. There are statistical methods to make everything valid and protect type 1 error. We can definitely do that. But the question is it going to improve power to detect drug effects? And is it still going to be clinically interpretable? And that's what the data will help us figure out.
I think it would be very helpful to hear from Jialu or David and Yolanda on the regulatory side of the statistics team. Your thoughts as we move into this new phase, which is really quite statistics heavy. Jialu or David or Yolanda?
Hi, thank you, James, for the very the great presentation. Well, from statistical point of view, I agree with you that once you start thinking about the composite endpoint, and there's a noise level that we cannot ignore. So once you start adding up different multi-organ components, there's a consideration of what will be the most useful component to be included, not just throwing everything into the composite. And also, I think I like the idea that separates the quality of life endpoint from the clinical endpoint. I think those two are addressing completely different questions. Also, there are some overlap, so it will cause some statistical complications, and we should separate them. But they both would provide some very useful information, so we should look at both.
Thank you. David or Yolanda?
I think we thought there was some good value in these composite endpoints, and there's so much of, I think the value in them allows you to get more patients into a single study, which means you could sort of make a decision quicker, which is what's needed in these conditions. And so I'm not sure you have to worry too much about what one dominating. If you define a good endpoint, as we've been talking about, I mean, obviously we've not decided what it is yet, but if you've got the time to response or the time to event, which are the sort of endpoints that James was discussing on his slides, then if different patients have different characteristics, then they can be a responder or fail by the characteristic that is dominating for them. So if they don't have a particular aspect, obviously they won't move on that, and they will move on other aspects that they have as part of their condition. And it does allow you to bring that in, and it'll just dominate on the way their natural condition is. So I think there's a lot of value in these. It does allow you to get lots of patients, which if you're just focusing on one endpoint, you'd have lots of trials with only a few people in because they've only got certain characteristics, and you might struggle to get anything from that with lots of very small underpowered trials, whereas these endpoints offer a lot of value, getting a lot of patients in there, making decisions more quickly. And you've always got the freedom afterwards, if you wish, to look at individual endpoints. They don't go away, but you can start off by getting something from this composite. So I think I see a lot of value in that.
Thank you. And thank you very much for this panel. I think in general, I think not just for this panel, but also for others, my sense from the group is that there are many details to discuss, but the general direction has been supported by the audience. I hope that's accurate. So I think it just really is time to step back and say a huge thank you. We very much appreciate all the work that was leading up to today and the great contributions today. Our next steps are clearly to consolidate the subgroup elements into proposed outcome groups, possibly ranked. We recognize that differences are likely between anti-plasma cell therapy and anti-amyloid therapy in doing this. And we also recognize the opportunity to consolidate some of the discussions with regulators on NT-proBNP as the surrogate. That seems very strongly supported here. I think we should also, there are several next steps that have pointed to data sets. So I think one thing we should point to right now is a thank you to those institutions providing such sets, though that will be critical to many of the next steps across the working groups and to the statistics exercise. And I think we'll also be thanking the working groups because some have a few intra-working group loose ends to tie up. But essentially in the next steps, the statistics team will be taking over a lot of the heavy lifting, as will, I think, specialized quality of life teams. And I think in that, we'll be looking also for more patient involvement as we develop different quality of life tools for AL. So a huge thanks again. And Preston, would you like to make some final remarks?
There we go. Sarah, can you hear me?
Yes.
Yeah, thank you. I'd just like to give everybody a humongous thank you for all of the effort of all of the people who have contributed to this effort. You know, I had begun my discussions with Isabel Lozada several years ago, actually, in developing this public-private partnership to tackle this. And, you know, if something's easy, it doesn't need this kind of global brain power to be brought to bear on it. This is hard, and it could not be possible to move something like this forward without each and every one of your contributions. I'd particularly like to thank the folks from industry and the MHRA for making the time to join this effort, because obviously we can't be doing this in a bubble in a vacuum in the United States without the input of the major centers, particularly around the world. And so the people who will be the recipients of that data at the regulatory agencies outside the US are just critically important to have them involved. And I really just want to thank you again, tell you how grateful I am that you all have contributed and joined us in this discussion. And it is going to be very interesting going forward. I do think that other disciplines are watching this group. I don't know of a consortium or a group that has gotten so far into the interstices of how to do clinical trials in a disease that's rare, more so than this one has. And so certainly what we come up with as both a process and an outcome, I think will be very helpful to other rare diseases.
Thank you. And I think it's the weekend for many of us. And I think those in Europe are even more so, but thanks again. Appreciate all the effort today.